This
giving me
conference will now be recorded.
this opportunity to share my story today. I will be talking about the humanized mice model for infectious disease, and especially in context with the viral diseases like dengue and HIV. So currently, I am a senior principal scientist in preclinical safety division in NIBR, Cambridge. And just before starting my presentation, just a disclosure that all the data which I will be showing today is generated within UMass Medical School with the support and the guidance with the Dale Greiner and Mike Braham, and a long and productive collaboration with Lenny Shoes from Jackson Laboratory. So the humanized mouse model, as we all know, there are three types of humanized mouse model. The first one, which I call the first generation model, is the one which is generated by transfer of a human PBMC. And this generation model is typically not used for infectious disease studies because of the very short study window period, as well as a lack of immune cells of different lineages. So the second generation and the third generation, which are produced, the second generation are produced by transplantation of CD34 positive cells, and the third generation, which is a BLT animal, which is generated by surgical implantation of fetal thymus and the liver, along with the transplantation of autologous hematopoietic stem cells, are the two models which are generally used to study the viral diseases. And most recently, the most commonly used is a BLT animal because of its more of its complete human immune system. And these two second and third generation models are currently used for various viral diseases, especially the human specific diseases for which there is no good animal models available yet. And some of these viruses are dengue, HIV, CMV, Ebola, etc. And then recently, which I called another mice, which is a fourth generation animals, where these either the HSC model or the BLT models are further transduced by a AAV, which expresses receptor of a specific virus, thus making them infectable with the respective virus. And this mice, which I am calling here a fourth generation, and I will talk a bit little about this mice in my later part of a talk. And this fourth generation is now have been shown to use for a COVID-19 or a SARS infection. So today my talk will be mainly focused on two human viruses, dengue and HIV, for which infects million in the worldwide, and there's no effective vaccines available yet. And the focus of my talk will be mainly the T cell and the B cells in respect to these viruses. So we started working on dengue viruses a few years back, and a typical experimental design for the studies will be as follows. So we used to take the humanized mice, CD34, that time it was CD34 transplanted humanized mice, and infect them with the various strains or serotypes of dengue virus. The viremia, we used to monitor clinical symptoms post-infection. Viremia was monitored by in the blood over the time of infection. The biodistribution of the virus was done by collecting different organs, and then we used to assess T and B cell responses, and finally we used to evaluate drugs or vaccines again in the efficacy studies. So upon infection, most of these animals used to get infected with the dengue virus, and the clinical symptoms were like weight loss, ruffled fur, hypomotility, skin rash shown in the top right. And these clinical symptoms were often strain dependent and as well as a donor dependent, and which is also seen in humans. So this pretty much recapitulated the human infection. And the severity of these clinical symptoms depended on the viral load most of the times. So here in the top right, we see a skin rash which developed in response to dengue infection. And then in the middle graph is the different strain of dengue virus, which infected at different level to these animals. And then when we do the biodistribution assay, we used to find that the viruses can be easily detected by simple PCR techniques in the liver, bone marrow, spleen, serum, etc. And all of these animals, upon infection, they used to generate T and B cell responses against virus. So this is a immunity and B cell response to dengue infection, and the figure in the left of the slide shows the interferon gamma response in response to dengue and, antigen, and at day 7 and day 14 post infection. And as we see, the T cell response, both the CD4 and CD8, in most of the animal have a very lower interferon gamma positive CD4 positive population or CD8 population, both at day 7 and as well as day 14. And the B cell responses, dengue specific IgM response were generated in these animals, but they were also significantly low compared to what we see in the wild type animals in general or the human. So this set of data suggested that both the T cells as well as B cell responses in these CD34 engrafted NHG mice were suboptimal. And one of the reasons we thought that time was that it might be due to defect in T cell due to inadequate education of human T cells in murine thymic environment. So next we proposed that if we engineer the NSG mice to express human HLA class 1 molecule that will allow human T cells to be more effectively educated intrathymically on human HLA alleles, and this will allow a more robust human T cell responses. So taking this hypothesis, Lenny from Jackson, he generated NSG mice by crossing not skid with the human HLA A2 positive animal, and so at the end he got the NSG which express human HLA class 1 molecules. And then we took this both NSG expressing A2 as well as NSG which is not expressing A2 molecule, and then we engrafted them with the same CD34 cells from the same donor. And then after 12 to 14 week post engraftment, once we validated the reconstitution of human cells, these animals were infected with dengue virus. And then the spleen was collected at week 1 post infection. And then we assessed the interferon T cell response mainly by intracellular cytokine staining in response to various dengue peptide pools. So this is a slide. So one major difference between the A2 plus and A2 minus mice was also in the numbers of the spleen cells. So in A2 minus animals, the spleens were relatively very small, and we used to get close to 3 to 5 million cells, whereas in case of A2 positive animals, the spleen was comparatively bigger, and the number of cells varied from 15 to 20 million. So it made us easier to set up numerous assays with the splenocytes. So here the graph in the right of the slide shows the difference in the response between the A2 minus and A2 plus animals. And as you see, there is a qualitative difference and as well as quantitative difference between the two strain. The A2 plus animals not only have a higher frequency of CD8 interferon positive cells, but there is also more numbers of peptide that responded compared to the A2 minus animals. And if you see the flow graph on the right of on the left of the slide, and if you see the pool B, we can see that there's a very well separated population of CD8 cells secreting interferon gamma in case of A2 positive cells, whereas in case of A2 negative animals, there is not much well separated cells were observed. And even with the positive control, which was anti-CD3, CD28 antibody, the good population of secreting cells were detected in A2 positive animals compared to A2 negative animals. So next what we did, we took some peptide which was A2 restricted. It was a dengue peptide, and it has shown to be to elicit a good T cell response in humans. So we took that same set of peptide, and then we did a similar intercellular cytokine assay in the A2 positive animals, and we found that the T cells proliferated significantly in response to A2 restricted peptide, and they secreted numerous cytokines like interferon gamma, TNF, and IL-2, shown in the right side of the graph. And these T cells were polyfunctional in nature. So this set of data suggested that the A2 NSG A2 plus animals are better, have a better T cell response in compared to NSG A2 minus animals. So while we are wrapping up the story, Mike Graham, who is again in UMass, he started generating BLT animals, and we started characterizing these animals. So BLT NSG animals have a higher level of CD45 compared to cord blood engrafted animals, and they have a lower. They also have a higher level of CD3 cells. Although there is the B cells are much more higher in HSC model compared to BLT. So we did the similar intracellular cytokine assay in the BLT and compared with the NSG A2 plus animals, and we found that in the BLT, the response in BLT animal are further enhanced compared to the A2 positive animal, and this response was very strong in compared to the non-BLT animals. So with all these sets of data, we suggested that the BLT animals are a good model indeed to study T cell response in respect to T cell response to an antigen. So what about the B cell response? So here I'm showing that dengue specific antibody response in a different animal model. And so for this, first we took the sera of a different dengue sera of the different model which were infected with the same strain of dengue viruses, and ran a ELISA, antigen specific ELISA, and we found that there was not significant difference in the antibody response between the A2 positive and the A2 negative animal, but the difference in the antibody titer was significant if we compared the BLT NSG over the NSG A2 minus animal. We also did the kinetics of a antibody response, and we found that the IgM response peaked at approximately at week 4, and however there was no IgG response at all in any of these models and at any time points. We also tried to see to evaluate that if we increase the viral inoculum into these animals, does it had effect on antibody titer? The effect was there, but it was very modest and not significant. And even when we increase the dosing frequency, the effect was not very significant again, suggesting that even though increased viral inoculum or a dosing frequency have a very small effect on antibody titers in these animals. So as I mentioned before, the only antibody generated in these animals were IgM. And although the antibody was IgM, but they exhibited the property which were very similar to what we see in a human, both via in vitro as well as in vivo assays. So the top panel is an in vitro assay, where in left side is the affinity is a measurement of affinity index, and here the antibody which were isolated at day 7 post-infection, which is an acute time point for a viral infection, the affinity index was far more lower compared to the antibody which was isolated at day 45, which is a memory phase for viral infection. The phenomena which is very similar what we see in the human patient. Similarly, these antibodies were capable of neutralizing dengue virus in a cell-based in vitro assays shown on the right. And this neutralization was dependent on the concentration of antibody. And then in the in vivo setting, these antibodies were able to provide protection to subsequent dengue infection, both in the prophylactic and a therapeutic setting. So the figure in the left shows where the animals were treated prophylactically, where animals were were left naive, or they were given a vaccine dose of dengue virus, and then when they were subsequently challenged with the pathogenic virus, the naive animal had a high viral load, whereas the immunized animal were having a very low viral load and were protected were protected and didn't exhibit any clinical outcomes. The figure in the right shows a therapeutic transfer study where the animals were first infected with dengue virus, and then they were passively transferred with the dengue antibody, where 3H5 is a dengue specific monoclonal, and the other two were immune sera, either neat or 1:4 dilution. And again, the animal which were treated with dengue specific antibodies have a lower viral load compared to the untreated animals, suggesting that the antibodies generated in these animals have the neutralization capacity similar to what we see in the human. So this portion of the talk, conclusion from this portion of the talk is that the T and B cells are generated in these models, at least in response to the viral infection. However, these responses are better in BLT, and then BLT, then NSG HLA2 positive animal, and then NSG. And the reason we think is that the improved education of T cells in contrast to human HLA molecule. The IgG response in all of these models are either is low or negligible, and this is due to ineffective class switching in these models. And this ineffective class switching is due to lack of secondary lymphoid structure, insufficient human cytokines and chemokines that drive the class switching, impaired B cell maturation, and finally the poor reconstitution of myeloid cells, mainly the antigen presenting cells like dendritic cells. And due to this, there's a lack of appropriate signal from dendritic cells to the naive T cells, which ultimately shapes the adaptive immune response subsequently. So we asked the question that is there's a better model with the improved human B cell development that permits the generation of antigen specific antibody. So taking this question forward, again, Lenny generated HGM3 animals. So HGM3 energies are the energy which expresses SCA3 extra additional cytokines, which is SCSF, GM-CSF, and IL-3. And all these three cytokines play an important role in hematopoietic stem cells engraftment, as well as the engraftment of the myeloid cells. And when Lenny generated these animals, Mike Graham took these animals from Lenny and he generated a BLT model. And then we characterized this model extensively. And as you see in the figure on the top panel, the human CD45 level of in HGM3 is significantly higher, at least in blood, compared to the NSG model. Although their levels are comparable in spleen and bone marrow, the human CD3 is less in HGM3 compared to the NSG model. And this, more importantly, human CD33 positive, which is a myeloid population, is significantly higher in both blood and spleen in HGM3 model compared to NSG. The HGM3 model also have a high level of FOXP3 T regulatory cells, and their T cells are less naive, which is shown that by the graph, two graph below, showing the lower frequency of a CD45 RA positive cells. And then the B cell in these animals, the frequency of B cells were comparable in both NSG and HGM3 in all three blood, spleen, and bone marrow. However, however, in HGM3 mice, the B cells are much more mature, and so some of the figures shown in the right of the slides shows that the immature B cells and the immature transitional and the transitional B cells are far more less in SGM3 animals compared to the NSG model. Whereas the naive B cells, which is CD27, CD10, and IgD positive, are the B cells which goes to the lymph node and then where they mature into the memory and the plasma cells, are significantly higher in NSG SGM3 animals compared to the NSG animals. And as a result, in SGM3 animals, due to the high frequency of naive cells, we see relatively higher level of memory B cells in the blood of the SGM3. The plasma cells, which is CD138 population, is lower as such in all the three, in both the model, in all blood, spleen, and bone marrow. So due to the presence of now the higher level of mature naive B cells, the overall antibody titer in NSG SGM3 animals are several fold higher compared to NSG animal, both the IgM as well as IgG. And then this correlated well with the dengue specific IgM and IgG response, where in case of NSG they were relatively low. In case of SGM3, at least the IgM response is very high, and we are able to detect some IgG response as well. So now I will change my gear a little bit and talk about the importance of a dendritic cells in shaping the adaptive immune responses. So as we all know, that dendritic cells play a very critical role in either providing immunity, protective immunity, or a tolerance. And the development of these two incidents depends on the three signals which are being presented to the naive T cells by dendritic cells. The signal one is the presentation of antigen in contrast to MHC class 1 or class 2. The signal 2 comes from the upregulation of co-stimulatory markers like CD80 and AX. And the signal 3 comes from the secretion of a pro-inflammatory cytokines like IL-12. And in order to have an optimal adaptive response, both the T cells and the B cells, all three signals need to be provided by dendritic cells. And these signals are only provided by mature dendritic cells. So the entire cycle starts by recognition of PAMs by the intracellular PRRs present in the dendritic cells, and this recognition leads to the maturation of dendritic cells, and these mature dendritic cells in return provide all these three signals to the naive T cells, which goes on and differentiated into different population of T cells and also affects the production of antibody. So if either one of these signals are missing, or if these signals are not present in a threshold level, the immune response become more tolerogenic over the protective immunity. So in contrast with HIV, all the HIV infected individuals have a significant level of antibody as well as T cells response. However, still the HIV persists for a long time, and eventually HIV overcomes the immune response and the individual suffers from AIDS. So there are few reasons why HIV maintain itself in a chronic state. One is that HIV mutate to evade the immune system. The second, it tries to maintain itself in a latent state in an immunoprivileged compartment. And the third, and which is more recent belief, is that the cell intrinsic sensing of HIV by dendritic cells are incomplete. Rather not optimal, as a result of which the dendritic cells is not able to provide all the three signals sufficiently to naive T cells, which ultimately result to a dysregulated immune response, as a result of which HIV continues to stay in the individual for a long period of time. So one of the reasons we think that the dendritic cells are not able to sense these HIV is that the dendritic cells express very low level of HIV receptor like CD4 and CCR5, and there are several cell intrinsic host restriction factors which prevent HIV from replication within dendritic cell. So how do we prove that? To prove this hypothesis, we took a pair of HIV virus called GRCSF and GRFL. These two viruses are identical. They are isolated from same individual, The GRCSF virus is a T-tropic virus, meaning that it infects the cells which have a moderate to high level of CD4 receptor, like T cells. Whereas the GRFL virus are an M-tropic virus, and it is capable of infecting cells which have even very low level of CD4 receptor, like dendritic cells. So before going into the in vivo system, we tested this in the in vitro system in the PBMC derived dendritic cells, and we found that the M-tropic, which is a GRFL virus, replicated well, whereas the T-tropic virus failed to replicate within dendritic cells. So now we know that these viruses have a differential replication in dendritic cells. So we tested this in an in vivo system. So we again took the humanized mice and we infected them either with the T-tropic virus or M-tropic virus. And then we monitored the antibody response, the T cell response, and then we finally re-challenged these animals for an efficacy study. So the T cell response shown in the bar graph, we found that the M-tropic virus, which is a red bar, the T cell response is significantly higher compared to the T-tropic viruses, which is a blue bar. And the effect was most significant for antibody response. Here, the red line is an anti-HIV IgG response, which we haven't seen before previously with such a high titer. So the MAC-tropic virus was able to elicit a very high level of anti-HIV IgG response, whereas a T-tropic virus, the response was very low, the level similar to what we have seen before. And then the antibody, which was generated in GRFL virus or M-tropic virus, have a strong neutralizing capacity, and it neutralizes both the M-tropic and the T-tropic virus at a very low IC50, whereas the antibody, which was isolated from T-tropic virus, failed to neutralize any viruses whatsoever at any concentration, suggesting that the M-tropic viruses are capable of eliciting stronger T as well as B cell response. So next we did the efficacy study where we took the same M-tropic and T-tropic viruses and we took a very small dose and attenuated it so that it becomes attenuated in replication and used as a vaccine. So we again gave these vaccine viruses, very small dose of vaccine viruses to humanized mice. And then after a booster dose, we challenged these animals with a high level of pathogenic HIV virus. And then we monitored the viral load over time. And what we found that in case of M-tropic virus, which is a red circle, none of the animals were infected even at week 8 post-infection, whereas in case of T-tropic virus, which is shown by the blue triangle, the animals started showing viral load by 4 weeks, and by week 6, most of the animals were infected with the virus. So we think that here in this study, we think that the MAC-tropic virus, which is a GRFL, because of its capacity to infect dendritic cells, can enter productively into dendritic cells, and within dendritic cells, the viral genome has been reverse transcribed and the cDNA is formed, which is recognized efficiently by pattern receptor recognizing receptors, and these leads to the maturation of dendritic cells, and these dendritic cells are now able to provide all the three signals sufficiently to the naive T cells, resulting in a better T and B cell responses. So next, I'm going to show you some data which shows that how this humanized mouse model can be used for dendritic cell-based immunization, which is also important in the field of cancer therapy. So here we use the BLT animal, but this can be also used for in an NSG model. So here we generated the dendritic cells from CD34 cell using the cocktail of cytokines, and then the immature dendritic cells were treated with the HIV antigen, and they were matured, and then this matured dendritic cells were given to the NSG animals, and after 3 weekly injections, they were infected with HIV. So the dendritic cells which are generated from CD34 cells have a surface marker profile very close to very close to the dendritic cells that are obtained from CD4 from PBMC, CD14 isolated from PBMC. They have similar levels of HLA-DR, CD11C, and then NDC signs. And what we found that when these animals, the animals which were immunized by a dendritic cell, they have a very robust T cell response, as shown in the figure in the left. It's an Alice spot in response to GAG and envelope protein. These are two immunodominant protein of HIV. And if you see the HIV groups, all the animals have almost 10 to 20-fold higher level of T cell response compared to the control group, which is 6 to 10. And these animals also have a significantly lower viral load or no viral load, shown in the black dot in the figure in the right. So most of the immunized animals have a minimal viral load, or some are even uninfected, whereas a control group does have a higher viral viremia, and they eventually show a much higher viral load by week 4, suggesting that the dendritic cell vaccination provides a strong protection to HIV infection. And we know that this protection is coming from HIV, from dendritic cells also, because when we did the same set of study, but here we didn't use it, and we immunized the animal without dendritic cells, the T cell response is significantly lower, and most of the animal was not protected. They exhibited the high level of viremia. So with that, the summary I want to so the summary of this presentation is that humanized mice are a valuable preclinical model for human diseases like cancer, transplantation, autoimmune, and viral infections. BLT energy is a better model to study human immune responses because it elicits better T cells and the antibody responses. However, in the pharmaceutical settings where BLT is not feasible, even the NSG animals expressing HLA molecule can be a better model to study immunotherapy. Now, NSG HGM3 might be an improved model because it have an improved human cells development, and it might be a better model to assess efficacy, safety, and tolerability of a vaccines, drug, and immunotherapeutic agent. Dendritic cells play a critical role in shaping the adaptive immune response, and in our settings, especially in HIV, dendritic cell-based immunization confirm better immune response and provide protection against lethal dengue HIV infection. So this was with respect to dengue and HIV. What about the recent COVID-19 infection? Is there any humanized mouse model? So yes, the answer is yes. So this is the paper. There was a paper which was published by J. Victor Garcia group sometime in 2019, just before the pandemic, and they generated a BLT model with the human lung. So basically, the BLT model consisted of human liver, thymus, and lung, and they showed that the human lung in these model upon engraftment it can have all human epithelial cells, endothelial cell, and mesenchymal shell shown in the figure in between, in the middle, and then these lung cells are capable of possessed all kind of human immune cells like CD11C, 68, CD56, etc. And when they use this model for human CMV, and they found that this BLTL model can be infected with the human CMV, shown in the figure in the middle in the yellow bars, and the animals were infected productively, and then after 4 weeks the level of infection goes down because these animals are capable of generating anti-CMV specific IgM response as well as a T cell response shown in the figure in the right. So this study suggested that yes, the BLTL model can be used for respiratory diseases like MERS or SARS or RSV. And then recently there's a paper which just came last month, and it's still in a review, and it is published by Richard Flavel's group from Yale, where he have used a Mr. G6 humanized mice to study the SARS infection. And in this paper, he suggests that this animal model can recapitulate all the pathology of COVID-19. And actually, Mr. G6 is the same as SGM3 animals, except that it also except that it is on the RAG background instead of NSG background, and it have a transgene for IL-6. And this model might also be very useful to study cytokine release syndrome because of the high level of IL-6 present in this system. So with that, I will close my presentation, and I want to thank you all for your attention, and will gladly take any questions or open for discussion.